Wanted someone to go back in time with me. This is not a joke. You'll get paid after we get back. Must bring your own weapons. Safety not guaranteed. I've only done this once before.
Hey, I've seen this one. This is a classic. This is where Ralph dresses up as a man from space.
What do you mean you've seen this? It's brand new.
Yeah, well, I saw it on a rerun.
It's a rerun.
It's just a jump to the left.
Hi, everyone, and welcome back to the online spinal cord injury research seminar series on this Thursday. I hope you all are well. Quick note, if you haven't seen Safety Not Guaranteed, Back to the Future, or the Rocky Horror Picture Show, I suggest you do all yourselves a favor and just watch them. You won't regret it. My name is Warren Alley from the University of Kentucky, and we are traveling back in time and doing a rerun of Caitlin Hill's presentation a few weeks back. A quick note, I did pay the internet bills and I've upgraded to the platinum package, and so we should be okay today. Moderating today is our good friend, Dr. Megan Detloff from Drexel University. Megan, thank you for joining us. Megan, it is all yours.
All right. Thanks, Warren. Hi, everyone. It's great to see you guys today. As Warren said, we're starting again. Caitlin Hill is joining us today from the Neural Stem Cell Institute in New York, from her home in New York State. But before I introduce Caitlin, I want to make sure that you guys know some of the things that we've got going on behind the scenes. There's a committee that is formed by people who are interested, grassroots, if you will, that is working hard to set up, make programming decisions and figuring out our transitions as we move forward into long-term programs for you guys in our YouTube online sphere. If you are really interested and you really like what we're doing, please hit the like button that's down below and don't forget to tell your friends about it. This is something that we do for you guys, and we want it to be as much you guys driving the content as we are in choosing it. So please send us suggestions, sign up if you're interested in presenting, and trainees, don't be shy. You can sign up too, or if you think you just want to try moderating, that's also something to do. So now I'm going to go ahead, and it's my pleasure to reintroduce Caitlin Hill. As I alluded to earlier, she's from the Neural Stem Cell Institute in New York State. And I've known Caitlin for a long time now, where from our graduate school days. We both were at Ohio State University. Caitlin did her graduate work with Jackie Bresnahan and Mike Beatty at Ohio State, and then moved to University of Miami and the Miami Project to Cure Paralysis, where she worked with Mary Bunge and Pat Wood. And so today, Caitlin's going to talk about her work looking at stem cells and how we can use them to it's a different title. Sorry. Enhancing the transplant survival and how cellular preconditioning can benefit. So Caitlin, without further ado, take it away.
Thanks, Megan, and thanks everybody for coming back to my talk. I hope things go a little bit more smoothly today. So I'm going to be talking about the work we've been doing to try and enhance transplant survival with the question of, is cellular preconditioning enough? And so last time things went a little awry, and I think it's because I forgot to give credit to these two little guys. So these are my two cats. This is Maddie, this is Yip. They have been around for a long time. Apparently they're now considered geriatric cats. So I will continue on with the presentation, and I look forward to all of your questions and comments. So in my lab, and we've recently moved, I was at Burke Neurological Institute, and now I'm at the Neural Stem Cell Institute, a little bit further up the road from Westchester. Now it's in Rensselaer, New York. And in my lab, we really have been focusing on 3 areas of research. One is developing and screening strategies to promote the survival of transplanted cells, and that's the work I'm going to be talking about today. But there's 2 other areas of focus in the lab. One is to look at the cellular changes that happen after injury and how they influence injury development and recovery. And I'm particularly interested in dystrophic axonal ending and their interactions with other cells and the environment. And another area in the lab that we've been working on is testing pharmacological approaches for enhancing tissue sparing and recovery of function following spinal cord injury. So I've collaborated with a number of people testing different therapies. While I was at Burke, I collaborated with Brett Langley and Anthony Suave, and we looked at the effects of nicotinamide riboside, and it showed some promising potential effects, especially when given before injury. So not so clinically relevant, but potentially a mechanism of approach. More recently, we're beginning to test the effects of optimizing the efficacy of a nuclear export inhibitor in collaboration with a drug company. And we're looking for postdocs for this project. So once COVID is not necessarily over, but perhaps less disruptive, I'll be looking to hire some postdocs. So if you like what you're seeing in terms of the research we're doing, please reach out to me. But for today, I'm going to talk about the work we've been doing on transplantation for spinal cord injury. And there are a number of different cells that have been transplanted into the injured spinal cord for testing their effects on recovery. Many of them have moved forward from the stage of this is my favorite cell and this is what my favorite cell does in my favorite injury model to moving forward into the clinic. I think that there's now 54 different studies in the clinicaltrials.gov database of cell transplants for spinal cord injury. And so the field's really moving forward to putting these into subjects with spinal cord injury or persons with spinal cord injury. But I think there's still a lot of questions that we don't know the answer to. In my lab, we've continued to work with Schwann cells, in part because Schwann cells are a cell type that we know a lot about. They've been studied for almost 30 years now, from the first studies that were done by Dick Bungay and Pat Wood and Ian Duncan, where they put Schwann cells into demyelinated rat cords and showed that they were able to myelinate, through to the recent phase 1 clinical trial, where they were shown to be safe and they had no adverse events. But for those of us who have been in the field for a while, it wasn't particularly surprising that alone they're not going to be sufficient. And this is true probably for many of the cell types that we're studying. A lot of the work on cell transplants have focused on how we can maximize their efficacy, what are their beneficial effects, and less has been focused on what are the potential negative effects of putting in the transplanted cells and other things that we need to counteract in order to maximize their efficacy. So while I was still a postdoc in Miami, we began to look at transplanted cells that had labels, and we saw that there was a decrease in the number of surviving cells over time. At the time, this was actually somewhat surprising. It was certainly unexpected, and we spent a lot of time trying to figure out, are the cells dying? What's going on? Did they lose their label? And I think through many studies, we ended up demonstrating that there is a decrease in the number of surviving cells over time. So you can see this here in terms of transplant volume, but we also quantified this seriologically. And we went on to show that the cells were dying by both necrosis and apoptosis, and it appeared to be occurring prior to 3 days. So this was an early loss of the transplanted cells. Beyond 3 days, the transplants looked the same as what we had seen previously, and there was long-term survival of the cells. So really what we're talking about is an acute loss of the cells within an early window following transplantation. In my own lab, we've gone on to explore this further, trying to use different approaches methodologically to try and, one, develop methods that are faster to quantify cells because stereology is very time consuming, and 2, create approaches that we could potentially screen different strategies. But what we're showing down here is that using cells that were expressing luciferase, we transplanted them into the rat spinal cord, and we imaged them over time using in vivo bioluminescence imaging. And we saw that there was a decrease in the cells over time, and then it plateaued. And this was happening prior to 2 days. So it looked like this acute phase of cell death was occurring prior to 2 days following transplantation. And when we quantified the cells in the spinal cord at 3 days or 7 days by measuring the number of GFP positive cells, we saw that the number of cells stayed the same. So this really told us that there was an acute window within the first 2 days following transplantation where the cells were dying. And so this creates a very early window that we can start targeting to try and improve the survival of transplanted cells and understand what might be the effect of this acute cell death. So in healthy cells, whether a cell lives or dies is ultimately the balance between pro-survival and pro-death signaling. And so in a healthy cell, the balance is tipped towards survival. When there's damage, the signaling pathways tip it towards death. So we can think about how can we counteract this death? And there's 3 principal approaches. 1, you could try and inhibit the inducers of injury. And in our early work, we tried to identify what some of these inducers might have been. We looked at, was it a result of mechanical damage from passaging the cells through the pipette? Was it the delay between collection and transplantation? Was it the fact that they were being transplanted in minimal media? And in the end, we were never able to account for more than 10% of the transplanted death. And so short of identifying all the potential inducers, it'll be a very difficult approach to try and block the inhibition or inhibit the inducers of cell death. And the consequence of incomplete inhibition is that the cells are going to die. So another approach would be to target pro-death pathways. So once the cell death has been initiated, can we block those pathways? And we know that the cells were primarily dying by necrosis compared to apoptosis, but both processes are involved, and we did some studies to target those, and we had some success. We're using a pharmacological agent that targets the calpains, which are a mediator of necrotic cell death. We were able to demonstrate that we could double the number of surviving cells, so this suggests that we can target pro-death pathways, but instead of having 85% of the cells dying, we got it down to 70%. That still means that the majority of the cells are dying. And so additional approaches are certainly required if inhibiting pro-death pathways is going to be used. So we began to think about how else we could do this. And another approach is to activate cell survival pathways. So rather than trying to block the death pathways, what if we prime the cells so that they have more survival capability before we put them into the cell? And so we didn't know what would happen if we tried this approach. And so the question we really wanted to ask was, does the activation of pro-survival pathways prior to transplantation improve transplant survival? And does this lead to enhanced functional recovery after spinal cord injury? So does tipping the balance back towards the pro-survival eliminate some of those potential detrimental consequences? And in thinking about how to do this, we really wanted to develop an approach that would be more amenable to clinical translation. And for me, that meant developing an approach that we didn't necessarily have to put in with the cells. Could we treat the cells prior to transplantation and then put them in the cord in this altered state that would allow them to survive better? So can we manipulate them at this phase as opposed to at this phase? And there's certainly lots of evidence in the literature that preconditioning is effective. It's been shown to be one of the most cytoprotective mechanisms in a variety of conditions. And what preconditioning is you mildly stress a cell, and this can be done by a variety of inducers, hypoxia, ischemia, heat shock. And what this does is it activates pro-survival pathways within the cells that allow the cells to adapt to the injury so that when a more severe injury comes along, they are protected. And one of the mechanisms that the cells use during preconditioning is to activate transcriptional pathways. So you can imagine, instead of just adding one growth factor, if we target transcription factors, we can activate a whole cohort of proteins, and maybe that will be effective. And not only can you do preconditioning by exposing cells to a mild injury, but it can be mimicked in a variety of ways, so we can study it experimentally. And this includes pharmacological applications and overexpression of transcription factors, growth factors, and cell signaling pathways. One of the mechanisms of preconditioning that's been shown to be particularly effective in other cell types and demonstrated to have efficacy even in the nervous system is hypoxic preconditioning. So this has been shown for embryonic stem cells in the ischemic rat brain, and for bone marrow stromal cells transplanted also into the ischemic stroke model. It hadn't been tried in the spinal cord. The studies where preconditioning has been used in conjunction with transplants in the spinal cord was a single study that looked at whether if you preconditioned mesenchymal stem cells, this could lead to protection of neural stem cells that were co-transplanted. And in this study by O et al., what they showed is that yes, if you preconditioned mesenchymal stem cells, neural stem cells were protected. So not only did the earlier studies show that it could have autocrine effects, this study showed that preconditioning might have paracrine effects. And so we decided to start by focusing on a transcription factor called HIF1 alpha. HIF1 alpha is the master regulator of the hypoxia response within cells, and it results in the transcription of a variety of genes that are involved in many factors that we thought could potentially be important in terms of what cells are missing following transplantation. So it's important for cell growth and fate, mitochondrial functioning, glycolysis and glucose metabolism, and oxygen transport. What we did was we transplanted Schwann cells into the spinal cord, and we looked to see whether they normally expressed HIF1 alpha. So shown in this Western blot here, you can see this is spinal cord injury alone. There's very little HIF1 alpha in the cord at either 10 minutes or 8 hours after the time of transplantation. And in this case, we're injuring the animals, we're waiting 7 days, and then we're transplanting the cells into the injured spinal cord. So normally the spinal cord has low levels of HIF1 alpha 7 days post-injury. When we add transplanted Schwann cells, the levels of HIF1 alpha don't really change. So it seems that normally Schwann cells fail to elicit an adaptive response that includes HIF1 alpha, and potentially this could contribute to why they don't survive very well. So in order to understand the studies moving forward, I think it's important to understand a little bit about HIF1 alpha and how it's regulated. HIF is a constitutively synthesized transcription factor in cells. It is normally also constitutively degraded because it's a rapid response transcription factor. It needs to be able to sense changes in oxygen level. And so it's constitutively synthesized, and then these enzymes, the PHDs, the prolyl hydroxylases, are a family of enzymes that are oxygen, iron, and 2-oxoglutarate dependent. They recognize HIF1 alpha, they hydroxylate it at a specific site called the oxygen-dependent degradation domain. Then Van Hippel-Lindau protein recognizes this hydroxylation, results in the subsequent ubiquitination of HIF1 alpha and proteasomal degradation. And so what this means is that we can manipulate this system to enhance levels of HIF1 alpha in cells. So under conditions of hypoxia, where we don't have oxygen, one of the key cofactors for the PHDs, or normoxia, where we inhibit the PHDs aren't hydroxylated, which allows it to dimerize with HIF1 beta, translocate to the nucleus, bind to its response element, the hypoxia response element, and elicit transcription of HIF target genes. We can also manipulate HIF levels in cells by mutating the hydroxylation site in the ODD domain, and thereby creating a non-degradable form of HIF1 alpha that can then go to the nucleus and result in HIF target gene expression. So the question we began to ask is, can we stabilize HIF in Schwann cells, and does this lead to transcription of HIF target genes? To do this, we used two different constructs. We used a construct in which the oxygen-dependent degradation domain of HIF, so the one that normally targets it for proteasomal degradation, was fused to luciferase. So for this construct, if we can see luciferase, it means that HIF is not being degraded. And we also use luciferase downstream of the hypoxia response element. In this case, if HIF's able to bind to the hypoxia response element and elicit transcription, we should see luciferase activity. And we look to see what happened when we expose the Schwann cells to either hypoxia or two different PHD inhibitors, one being DFO or deferoxamine, which is an iron chelator, and AQ or adaptoquin, which was identified from a screen of compounds for their ability to stabilize HIF using ODD-Luc assays in other cells. And what you can see here is that for all three conditions, we were able to increase the amount of luciferase we were able to detect. So HIF was stabilized, it maximized, and then it created a steady state. And we saw the same with DFO and AQ. It's worth pointing out that if we look at the axes here, that the level of luciferase activity with DFO was roughly double that we achieved with hypoxia and adaptoquin, suggesting that it results in a more robust stabilization of HIF than the other two conditions. And then we look to see, well, is it able to transcribe HIF target genes through using the HRE luciferase construct? And what we found was that yes, indeed it was, and that all three conditions resulted similar levels of luciferase activity from the HRE. And so this shows that we can stabilize HIF using these treatments, and that it results in HIF target gene activation. And from these assays, we were able to establish the time frame for our future studies in terms of duration of exposure. And so the numbers highlighted in red are the ones that we went on to use. And we picked these time points not because they were necessarily the maximal time points, but because they resulted in robust stabilization and transcriptional activity of HIF, and were amenable to potentially being incorporated into a transplantation paradigm. So we used 24 hours of exposure to hypoxia, 24 hours exposure of DFO, and 16 hours exposure to adaptoquin. We then went on to confirm that we could see that HIF was stabilized with these treatments using Western blotting. And so here we're showing that we can see increased levels of HIF-1 alpha on the blots, and we quantified it. It was more stabilized with the pharmacological inhibitors of HIF than it was with hypoxia, and this is due in part to how we collected the cells. Rather than immediately scraping the cells, we're collecting them as we would for transplantation, so we have a sense of what those levels are in the model that we plan on using. We went on to look to see how which HRE target genes might be induced following stabilization of HIF using QIAGEN hypoxia arrays, and we showed that there were at least 8 different HRE-dependent mRNAs that were induced by the treatments, and they all induced them to a fairly similar level. So for most of the remainder, when I'm talking about demonstrating that the treatment's working, we focused on using VEGF and enolase as examples of HIF target genes. So using Western blotting, we confirmed that not only did the treatments induce HIF target gene expression at the mRNA level, but it also induced expression at the protein level. And so all 3 treatments are able to induce VEGF and enolase. And again, the pharmacological agents seem to induce a more robust expression of the HIF targets. So at this point, I'm going to stop for questions.
Hi, Caitlin. There are several questions coming through, and you guys in the audience, please keep your questions coming. So we'll highlight a few here. Our first is from Kathleen Cole. Kathleen asks, if you transplanted cells and then waited 1 to 2 days and then did a second transplant, so transplanted more cells, do you think the second cohort of cells would show better survival? And then she clarifies that by saying, do you think the initial transplanted cells are changing the environment of the lesion site at all?
That's a good question. I'm not sure anyone has done that. I think they're possibly changing the lesion environment, and we have some data coming up that will show how they might be changing the lesion environment. I'm not really certain. We haven't thought about doing it because our focus has really been on clinical approaches, but it would certainly be a question worth considering.
Yeah, it's something interesting. I mean, I follow regeneration stuff, but I haven't, I don't follow it as closely as you do, but I don't recall anybody transplanting twice.
Mm-hmm.
So that's an interesting one. Our next question is from Lana Zuludeva, who's at the Gladstone Institute. And Lana is asking, hold on, I have to find it. I just lost it. Okay. Have you seen a correlation between the dose or the number of cells transplanted to the amount of survival?
Yeah. So when we did the first experiments looking at transplant survival, that was the big question, right? Everyone was like, oh, you're putting in 2 million cells. That's too many cells. And so the experiment that we did is we transplanted 250,000, 500,000, 1 million, and 2 million cells. And then we counted the number of surviving cells at 10 minutes, 6 hours, 7 days, and 28 days for all of those. I think there was at least 8 animals per group. And the answer to that question is no. Regardless of how many cells you put in, the percent survival was the same, around 70 to 80%.
Wow, that's a lot of work.
Yeah, that was the year as a postdoc I spent 800 hours counting on Stereo Investigator.
Wow, that's impressive. Our final question of this break is from Peggy Asink, and she's asking, have you looked at whether the cells survive better when transplanted into females compared to males? Sex is a biological variable question for you.
Hi, Peggy. No, we haven't actually. I haven't done any transplants into males. Everything we've done has been into females. There was some work done by Damian Pierce in the early mid-2000s, where when we were trying to identify labels of transplanted cells, and they were using male cells into female rats. And I believe that percentage of survival was about the same, but the opposite hasn't been done. So female cells into male rats, I'm not aware of.
All right, awesome. We'll continue on, and we'll get more questions at the end in the next break.
Okay, great. So having demonstrated that we could stabilize HIF in the Schwann cells, and that resulted in expression of HIF targets, we asked, is stabilization of HIF-1 alpha induced by hypoxia or pharmacological inhibition of the PHDs cytoprotective for Schwann cells? And so to do this, we used an in vitro assay. We grew the cells, we collected them, we plated them into 96 well dishes. We let them grow for a set time, I believe 2 days. We then applied the drug, waited the specified times, and then induced cell death by applying hydrogen peroxide. And for this experiment, all cells were growing for the same amount of time before the assay. So we staggered when we applied the drug, when they were applied at different times. And what we found was that when we applied DFO and adaptoquin, that we got a large increase in the survival of the cells in vitro and in response to hydrogen peroxide. And that it occurred across a range of hydrogen peroxide concentrations. Interesting though, when we applied hypoxia, it wasn't protective. So the mechanism by which they're working is probably slightly different, and we've begun to explore this. So I put that out there, but I'm not going to talk about that further here. In any event, it appeared that from this experiment that DFO resulted in the greatest protection of the Schwann cells. And so we went on to look at what happened when we transplant the cells. And so for our transplant experiments, what we're doing is we are injuring the animals. In this case, we're using a 200 kilodyne contusion injury. In some of the later experiments, we're going to use a 250 kilodyne injury. Both are moderates. We did it for different reasons, but we specify which injury model in the figures going forward. We wait 7 days, we collect the cells, and then we transplant 2 million cells into the injury epicenter. And then we waited for different times. So 10 minutes, 3 days, 7 days, or 28 days following transplantation, and we assess survival either by histology or Western blotting. And so the first piece of data that we got in this pilot study of 3 animals per group at each time point was at 28 days, and it looked like there was going to be a doubling in the number of surviving cells. Unfortunately, when we went to look at the rest of the time series, what we found is that there was no difference in survival. And so even though we were able to elevate HIF within the Schwann cells, it doesn't appear to be sufficient to protect the cells. And this was confirmed using Western blotting, where we also saw that there was no difference in the amount of GFP within the cords at 7 days post-transplantation with cells with or without TFO. So this suggests that we're able to elevate HIF in Schwann cells. This results in the expression of target genes, but it doesn't seem to be sufficient to protect the cells. But as I mentioned earlier, one of the other studies showed that when you elevated HIF in mesenchymal stromal cells and then looked at the survival of stem cells, that it was protective, having paracrine effects. So we looked to see whether there were some paracrine effects of transplanting the cells. And we looked to see, here we're showing 3-nitrotyrosine staining. And so there seemed to be a reduction in nitrotyrosine early after transplantation. So this suggests 2 things. One, it suggests that the transplant itself may elicit some additional damage to the spinal cord, which we need to be mindful of. And 2, that pretreating the Schwann cells with DFO seemed to reduce this. So there might be a paracrine effect there. And then the other thing that HIF does is it's an important molecule in terms of regulating proteins important for blood vessel formation and oxygen regulation. And so we looked to see if there were changes in blood vessel formation, and we saw that there was a slight increase in the density of blood vessels within the injury site. So this suggests that even though the treatment wasn't able to protect the Schwann cells, it had some paracrine effects. And so this led us to ask, well, maybe this might be sufficient to lead to some protection. And so we did a more complete study. So in this case, we're doing a study that has at least 12 to 15 animals per group upon completion that was blinded and randomized. And we did the injury. Now we're using the 250 kilodyne injury, transplanted the cells at 7 days, and injected either medium, in this case, that's DMEM-F12, which is a minimal media, Schwann cells treated with hypoxia, Schwann cells preconditioned with DFO, or Schwann cells preconditioned with adaptoquin. And then we looked at behavior over 8 weeks and assessed histology at the end. And what we found is that there was no improvement in survival. So similar to the short-term study, the long-term study did not show an effect on survival. And it also didn't show an effect on behavior. And so it seems as though preconditioning was not sufficient to either protect the Schwann cells or to promote functional recovery. So this suggests that our hypothesis of preconditioning and the activation of pro-survival pathways might not be sufficient in order to protect the Schwann cells or the transplanted cells. And so the consequence of incomplete activation is that the cells die. So what we haven't shown is it that activation of HIF is insufficient for cytoprotection, or is it that the adaptive response induced by preconditioning is too short-lived? And so before I continue with those experiments, I'll take some additional questions.
Okay, great. The questions just keep coming fast and furiously, Caitlin. So we'll ask a few. Our first is from Inez Maldonado. Inez asks, have you looked at the effect of priming in survival genes or priming survival genes such as BCL2? And her second question is, do you see effects on inflammatory pathways in the spinal cord or in the cords where you've put in these preconditioned cells?
So we haven't done any experiments with BCL2 to try and block death that way. And the second one was the
Asking about
inflammatory pathways.
Yeah.
We have not looked at inflammatory pathways in the cord directly. We did collect some cords and do some of the hypoxia arrays on them. But the magnitude of the gene changes following transplantation were not particularly large, I seem to recall, and so we haven't looked at that any further.
Yeah. Okay, great. Our next question is from Beatriz Martinez Rojas, and she's asking, HIF-1 alpha is thought to also be induced in the endogenous cells of the hypoxic environment of the lesion.
Mm-hmm.
It was sorry, that was a question. HIF-1 alpha or HIF-1 thought to be induced in the endogenous cells in the lesion.
So in the first Western blot that I showed, that was actually after injury. So we did spinal cord injury and looked at HIF-1 alpha expression at 7 days post-injury, which was the time we were transplanting. And then we added the Schwann cells and looked to see what the HIF-1 alpha levels were. And in both cases, the levels of HIF-1 alpha were low. So it doesn't seem to be a transcription factor that's expressed at high levels in the cord 7 days post after injury when we're transplanting. That's not to say it's not at earlier time points.
Okay, and then our final question of this break is from Zin. Sorry, Zin, I'm going to butcher this. Kaying. And Zin's asking about your thoughts on preconditioning the cells versus making transplanted cells happy by including growth factors or matrix molecules, especially on cell integration and function.
That's a great question. And I think I'm going to punt that to the end of the talk.
Okay. Sounds great. Carry on, Caitlin.
All right. So we showed that we could elevate it. We showed that we could induce HIF target genes. We showed that it was insufficient. So then we began to ask, well, why was it insufficient? And so the first question we're going to address here is it that HIF is insufficient for cytoprotection? So maybe HIF's the wrong target. So to do this, we expressed a retroviral construct of VP16, which HIF in the Schwann cells, and we looked to see, one, that we could elevate HIF with this construct. And so the Western blot shows that we can indeed elevate HIF using the VP16 HIF construct, and that it results in roughly a twofold induction of the two representative HIF target genes. We then went on to test this using an in vitro assay, like I showed before, where we put the cells in the dish, we induced cell death, and then we assayed to see the number of live cells. And you can see that when we express VP16 HIF in the Schwann cells, we're able to induce some protection. But if we compare that to what I showed earlier for the pharmacological inhibitors of HIF, you can see that the protection with HIF alone is less than what we are achieving with the pharmacological inhibitors. So it seems that the pharmacological inhibitors do induce a more robust protection than HIF alone. And that's likely because HIF is just one of approximately 20 transcription factors induced by hypoxia. And there are also at least 10 transcriptional repressors induced by hypoxia. So it's probably only one component of hypoxic preconditioning. We then went on to transplant the cells that overexpressed HIF in the Schwann cells to see whether this was protective. And so we're using a 200 kilodyne injury, waiting 7 days, transplanting 2 million cells, and we're comparing Schwann cells, VP16 Schwann cells, and VP16 HIF Schwann cells using a blinded experiment and that was randomized. And what we found is that we were indeed able to elicit protection of the Schwann cells. So HIF itself is able to protect Schwann cells against transplant-mediated cell death, although it was not as robust as we would have liked to have seen. But it does at least show that HIF can protect the cells. And so it is unlikely that the lack of protection with the pharmacological inhibitors is because HIF isn't protective. This led us then to ask, well, if HIF can protect the cells, is it that the adaptive response induced by preconditioning is perhaps too short-lived? And I'll pause for a second in case there were some questions on the genetic experiments.
I think you're good, Caitlin. Go ahead and keep going.
And so we then asked, well, is the effect too short-lived? And so for this experiment, what we did is we injured the animals, we waited 7 days, we transplanted the cells, either Schwann cells alone, hypoxic preconditioned, pretreated with DFO, pretreated with adaptoquin, and then looked 10 minutes, 6 hours, 24 hours, or 48 hours after transplantation to see what the levels of HIF-1 alpha were, and whether HIF target genes were continued to be expressed. And so here is the Western blots, and what you can see is that with hypoxia treatment, 10 minutes after injury, there are elevated levels of HIF, and this holds for the pharmacological inhibitors as well. At 10 minutes, there's increased levels of HIF here and here. At 6 hours, the levels are lower, and by 24 hours, it's back to baseline. And so HIF-1 alpha levels within the cord following the preconditioning of the Schwann cells rapidly return back down to baseline levels within 24 hours after transplantation. And we found the same result when we looked at the expression of VEGF mRNA. It was initially elevated at 10 minutes and 6 hours with all 3 treatments, but by 24 hours, it was back down to baseline. So the adaptive response that we're able to induce with all 3 of the treatments is short-lived, which brings us back to our previous picture where we're looking at the time frame in which cell death is happening. And we wanted to target a relatively early time window, but even this 2-day time window seems to be too long for preconditioning to be effective. And so in considering how we were developing an approach amenable for clinical translation, we were really trying to focus up here and how we could manipulate the cells prior to transplantation, but perhaps it's not enough, and perhaps we need to be considering what to do and what to put in along with the cells. And so in a final series of experiments to try and understand, is this approach feasible and will it work? We asked, does the addition of post-conditioning augment survival? So can prolonged inclusion of the pharmacological stabilizers be protective? And so we have some in vitro data to suggest that this might be the case. Again, here's the preconditioning treatment results I showed earlier. And when we added the post-conditioning, so here we are, we're adding the cells, we're pre-treating them, and then we're adding the drug in again at the same time as we're inducing cell death. We see that for DFO, there's not really a very much of a difference, but for adaptoquin, the addition of post-conditioning further augmented survival and basically brought it to the levels that we were seeing with DFO. So this suggests that in some instances, the inclusion of pharmacological agents along with the cells may be an approach that needs to be considered. So we then did another animal experiment where we injured the animals, we waited 7 days, we transplanted 2 million cells, and we transplanted either Schwann cells pre-treated with DFO, or Schwann cells pre-treated with DFO plus inclusion in with the transplantation media. And then we looked at survival of the transplanted cells. And unfortunately, this too was not protective. So it doesn't seem that adding DFO just in with the cells is sufficient to protect the cells. So we did another experiment where we then did the injury, we transplanted the cells, and this time we mixed the cells with a matrix. We used Matrigel, a minimal growth factor Matrigel, which Damien Pierce had previously shown could be protective for Schwann cells. And we transplanted Schwann cells with Matrigel, Schwann cells plus DFO mixed with Matrigel, and Schwann cells plus DFO, Schwann cells with Matrigel, Schwann cells pre-treated with DFO and then mixed with Matrigel. Schwann cells pre-treated with DFO and then mixed with Matrigel that also included DFO. And again, it was insufficient to protect the cells. So at least for the pharmacological manipulation of HIF using DFO, it doesn't seem that even including it along with the cells at the time of transplantation, either when the cells are mixed with just media or mixed with a matrix, seems to be sufficient. We haven't gone on to do the Western blots to show whether HIF levels were elevated in the cord or whether the target genes were elevated for more prolonged periods of time. But it seems, at least for DFO, that this approach will not be sufficient. So this leads me to conclude that preconditioning and short-term activation of HIF is insufficient to protect cells following transplantation. And so we've tested this hypothesis now. I'm fairly convinced that preconditioning for transplants and spinal cord injury is probably not going to be sufficient, and that we need to be thinking about what we can include along with the cells at the time of transplantation, which kind of gets back to the question that I was asked that I punted. So while I've been working on preconditioning, several other labs have made tremendous strides in terms of understanding what might need to be included along with the cells. And so Mark Tazinski's lab, along with Paul Liu and many of the trainees that have come out of his lab, have gone on to show that if you include a 4-factor cocktail, which includes BDNF, basic FGF, VEGF, the calpain inhibitor that I initially showed, along with fibrin, that you're able to get very good transplant survival. And they're showing now some really nice work being done about what can be done and what we can understand about the impact of transplants now that we have better survival. And then just last month, Giles Plant and Sarah Helshorn showed using a new suspension material that if you put Schwann cells in the shield suspension media, that it is able to increase the survival of the cells. And so this now suggests that there's different approaches that can be used as we think about moving forward about what is it that cells need to be included with along, what needs to be included along with the Schwann cells at the time of transplantation. So from the time I started this project, which was a spin-off project that started with a collaboration with Lawrence Moon when I first started in the Bungie lab, the question I really want to know is, does this death matter? Does this early death of transplanted cells limit functional benefits of cellular therapies? And I think that we're beginning to understand the answer to this, and I think the answer is yes. So again, in this recent paper by Giles Plant and Sarah Helshorn, what they showed is that when they had the animals that had better transplant survival, they made fewer missteps on the horizontal ladder walk. And so it does seem that just improving survival may be enough to counteract some of these deficits. And so we've worked tirelessly on trying to understand this problem. There were many people who worked on this project along the way. There were many experiments that did not turn out as expected. And so I would like to thank all of those people who have helped out along the way. We're moving on from this, taking what we learned about transcription and transcription factors and moving towards new projects in the lab, which we're excited to be starting. But I think that the results from these studies are really important because it suggests that, you know, we, instead of thinking, oh, we can manipulate the cells before we put them in, we really need to be thinking about what is it that we have to put in with the cells in order to maximize their efficacy as transplants move forward into clinical use for spinal cord injury repair. And so, again, I'm looking for postdocs for the new projects in the lab. Feel free to email me. And I was once young like my kitty cats when I started this project, and now I feel a little bit like, yep, putting my hand over my eyes about, oh no, him. And so one last slide and I'll leave up while I take additional questions. I mentioned this the last time, but at the Neural Stem Cell Institute, Neurocell is a spin-off company, core component of it where you can get iPSCs and different cell lines. They do different work. So if you see anything on this list that might be relevant to the work that you're doing, feel free to reach out to them. They'd be happy to help you with your studies.
Hi, Caitlin. That was really excellent. There are a lot of questions, and we'll start off with one from Abel Torres Espin. And Abel asks, what if the major factor is external to the cell? Host versus graft reaction can happen even in some autologous transplants. Have you looked at immunosuppression combined with the HIF, VP16 HIF?
We have not done immunosuppression along with HIF. So in the first experiments that we did with cell transplants where we were looking at transplanted cell death, we were using cells from transgenic rats that expressed alkaline phosphatase, human placental alkaline phosphatase. And one of the reasons that we failed to identify our cells is because that's a cell surface receptor. And those cells underwent 2 phases of cell death. They underwent an acute phase of cell death, like I'm talking about here, and then they underwent a second phase of cell death that was immune-mediated and rejection. And when we applied cyclosporin, we were able to rescue the second phase of cell death, but not the first. So that's not to say that there's not an immune component to the early cell death, but it's probably not the largest factor.
So Jerry Silver from Case Western Reserve has asked a question towards the beginning of your talk that kind of has followed through from some of the other questions. And so we'll start with Dr. Silver's questions and comment. He said, I would have thought that the transplanted Schwann cells might be proliferative. So why after the cell death phase do they not divide and amplify their numbers?
So that's probably the only one little bit of data from Mary's lab that we never published. Although I think Damien did publish some on proliferation, maybe. But I had an undergrad who looked at proliferation, and very few Schwann cells proliferate following transplantation. So when you transplant them, they've been growing in proliferative media, and so early, I would say we probably looked at 10 minutes, 6 hours, 3 days, 7 days. 10 minutes, 6 hours, there's still some proliferation. It's less than 5%, I believe. Or there were no more than 5% certainly at 7 days. It was a very, very small percentage of cells, I think even less. Because once Schwann cells make their interaction with axons, if there's sufficient Schwann cells, then they're able to myelinate, right? So normally for Schwann cells to myelinate, they come in contact with axons and then they proliferate and then they myelinate. But when there's sufficient Schwann cells, then they don't need that extra proliferation phase, it appears, based on the quantification that we did. And that was looking at MCM2 as a marker.
Kind of to follow up on that question, Sasha Rabchevsky from University of Kentucky has asked a series of questions, and he starts with, I understand the history and clinical use of Schwann cells, but can you expect to see anything to improve survival that aren't supposed to be there normally? Would you expect the same results if you used fibroblasts?
Yes.
And
so,
then
there's like, there's a lot of people who don't necessarily think Schwann cells are the best cell type to be transplanting in the cord. I think that we don't know what the best cell type is. I think we know a lot about what Schwann cells can do and what Schwann cells can't do, which makes them a good cell type for answering these types of questions. And we know that they can promote axonal growth and can have beneficial effects. So the cell death I'm talking about is generalizable to all cell transplants. Okay. Actually, Schwann cells survive slightly better than stem cells. So in the work that's been done on stem cells, the survival rate of stem cells tends to be around 5%, and for Schwann cells, it's around 20%. I don't know that anybody's quantified the survival of fibroblasts.
I'm just getting feedback from Warren. So Warren would like to like me, and I'm multitasking on multiple computers here. So Paul Ryer is asking, and he has made a comment here, and so we are shifting gears and having Paul on this and talking. We're really excited. So here's what Dr. Ryer asked. So he says, schwannosis has been described in human spinal cord injury pathological specimens at the Miami Project. Any thoughts on why endogenous cells do better in the injury environment? And are there rat versus human or exogenous versus endogenous sources?
So I think so I'm not entirely clear on what Paul is asking, but yes, schwannosis occurs, and even following transplantation, and we showed this in the 2007 paper, that the transplant is actually a mix of the cells you transplant and host Schwann cells. And so we looked and we compared that, and I forget what the proportion was, but host Schwann cells and grafted Schwann cells are what end up making up the transplant. I don't think I answered Paul's question though.
So Michael Lane actually sent me a question via text and because he couldn't get it to type in. So I'm going to read off of my cell phone right now. Michael is from Drexel, and he's asking, what contribution might the recipient species and strain make to donor cell loss? Some reports suggest even Fisher Schwann cell survival in Sprague Dawley, if there's CSA immunosuppression, might be better than survival in Fisher rats. Can you?
Yes. So I think that this is actually a really important point, Michael. So we know that Fisher rats are so highly inbred that rats from one barrier are different from rats from another barrier. So all of these experiments were done with cells from one barrier transplanted into rats from that same barrier, because there were some studies that were done in Mary's lab that did show that if you had cells from one coast going into rats from another coast, that the survival was different. But all of the studies I'm doing, they were all from the same barrier, which then created problems when the company decided to redo that barrier.
It's very interesting how that goes, isn't it?
Yes.
So we have another question from Reza Jalili, and Reza is asking, have you looked into transplanting cells into non-injured spinal cord and following cell survival?
Yes, we did that. They still die. They probably die in greater numbers. But yeah, that was one of the things that we did, is we injected them into the uninjured cord to see if we could see them in the earlier experiments where we were trying to figure out why we couldn't find our labeled cells.
And then just a couple more questions. So Victoria Moreno is asking, did you compare the intramedullary injection versus intrathecal delivery in CSF or within a hydrogel over the lesion without inducing additional damage by the injection itself?
So Schwann cells work because they provide a good substrate for axonal growth, and they're able to remyelinate. And so giving them above the cord or injecting them intrathecally, we don't think that they would do what their primary functions are. So no, we haven't done that.
Yeah. And then I think we'll close up with a question from Liz Bradbury. This was from a little bit earlier in your talk, and she was asking, bioluminescence seems great for longitudinal assessment of survival. What resolution can it give for more dispersed cells? For example, if the transplanted cells migrate, would they still be visible?
So I think in vivo bioluminescence is a great tool for studying things like cancer, where the tumors are growing. It is a less good tool for studying transplanted cell death. We spent a lot of time trying to optimize it. We were able to show that we could use it to see the decrease in the number of cells, but we were never able to use it to see really differences in survival. So you'd have to have something that was really, really robust in improving survival in order to see it, I think, in the spinal cord. There's a lot of technical problems in terms of depth. There were some early studies, Rutenberg, I think et al., where they had some success in mice, and the Japanese groups have had some success primarily in mice, but it's very difficult to do in the rat. And I certainly won't be continuing to use it. So, but if anyone wants to try it, I'd be happy to tell you more about it.
All right, great. Well, thank you so much, Caitlin. I'm sure there's tons of virtual applause happening all over the world for this, and congratulations on making it through.
I know.
On our YouTube channel. We really appreciate you being such a good sport about this. As a note, I want to acknowledge our program on Tuesday. So on Tuesday, we're excited to have Dr. Chet Moritz from the University of Washington as our featured presenter. And the title of his talk is Stimulation of the Spinal Cord to Improve Forelimb, Hand, and Arm Function After Cervical Spinal Cord Injury. If you haven't had the opportunity to hear Chet give a seminar, I highly encourage you to join us and tell your friends, because it really will be an excellent presentation. And it will be translational. So as you can guess from the title, there will be some animal work and also some human work as well. So please join us on Tuesday. Thank you again to Caitlin. Caitlin will hop over to YouTube if there are any more burning questions. She'll be there for a little bit. We'll leave it up. And be sure to sign up, hit like and subscribe, and tell your friends. And we'll see you here next time. Thank you very much.